Okay, so thank you very much for inviting me to present our project that was supported by the CJD Foundation this year. And I will start by thanking all the families and I will say the whole CJD Foundation for supporting our research throughout the years. A little bit of background of where we are. We are in the northern eastern part of Italy, nested inside the UNESCO World Heritage Dolomites, these fabulous mountains. Our center is characterized by the definition of cross-disciplinarity, because in reality, what we have is lots of different techniques and lots of different expertises brought by the faculties that come together to make a true cross-disciplinary science that try to break the barriers between disciplines. And I hope that what I will present today will provide an example of this concept. So my lab has been active now for several years on a single protein. I keep saying that I became monomaniac in my career because the reality is that I just study one single protein. That is, this protein probably, quite familiar with all the audience that are hearing us today. The name is the Cellular Prion protein. This is a protein that usually resides outside cells, in particular nerve cells, on the surface of these cells. Unfortunately, we still don't fully understand what this protein does in the brain, but we actually understand something, let's say, dark about this protein, its ability to eventually change its form, something that in biochemistry we call conformation. Well, this protein is able to basically change its form to become something else, something actually toxic and dangerous, something that then polymerizes and makes this sort of structure that you can see here on the right of the slide that we call amyloid fibrils. And these amyloid fibrils eventually accumulate in the brain of patients affected by prion diseases and, of course, create a very devastating kind of phenotype that we all know. So historically, in the prion field, people have been working on the idea of trying to clear up these amyloids from the brain of patients. Eventually, this concept got a little bit old-fashioned when we realized that this was very difficult, if not impossible to do. In parallel, another concept started to emerge. That is the idea of actually targeting the normal form of the protein, so the cellular prion protein that I introduced a few seconds ago, the protein that we all have on the surface of our nerve cells. Targeting this protein pharmacologically in various different ways would actually represent a way, an opportunity for blocking the formation of those toxic amyloids at the very beginning, because we will actually stop that misfolding process at the very beginning. And without taking too long, there is clear evidence in the literature that we can actually block the expression or actually block the function or both of these protein without damaging, without creating any damage to the nerve tissue, eventually to the human being receiving the treatment. In the literature, especially in the data collected in laboratory animals, we know very clearly that these kind of concept of blocking the expression, so reducing the loads, of abrogating completely the expression of this protein in animal models, have a tremendous impact, beneficial impact of the disease on the disease. Just to make an example, if we create mice that are completely knockout, meaning that they don't express at all the cellular prion protein, they are 100% resistant to any prion disease. And the same will very much be true in human. In fact, this is actually an image that we published with a few colleagues a few months ago to try to rationalize ways that we could try to explore in order to obtain this effect, meaning reducing the loads of this normal protein in the brain of patients. And one immediate intuitive way is pursued by Ionis Pharmaceutical, which is trying to develop so-called ASOs, or antisense oligonucleotides. These are compounds, are let's say non-canonical drugs that actually impair the normal production of the protein at the very beginning again, and eventually then reducing the loads of the protein in the brain. And this is definitely a very promising approach. What my lab is trying to do is something that we consider complementary to what Ionis is doing. We want eventually to try to answer to this question. Is there a way to selectively suppress Prpc with canonical drugs? By canonical, I mean small molecules, so drugs that, like many others that are in the clinics for treating many diseases. And okay, in order to explain what we're trying to do, I will start very easily to recapitulate how a protein is made in a cell, in whatever cell you may imagine. In fact, what you see here is called the central dogma of biology, and is basically transmission of information, an information encoded inside the DNA that then gets transcribed into a molecule that we call RNA. This RNA is processed to make a messenger RNA, and the word message means that then this molecule travels to other kind of structures in the cells that are called ribosomes, and thanks to the information encoded into the mRNA, the ribosomes basically print proteins by assembling one amino acid at a time. And the endpoint, the result of this translation of the information encoded into the mRNA, is basically a long chain of amino acids, a unique chain, unique for each protein. Now, there is another process, very important here. This chain of amino acids that you can see here then fold on each other, or each amino acid on each other, to specify that form of the protein that I referred to as, a few slides ago, and then makes the final form of the protein, for which, thanks to which the protein then exerts its biological function. Now, we are particularly interested in this process, this folding process. It turned out that this process is very complicated, because you can imagine that some protein may actually be made by, several hundred amino acids. Our protein, the cellular prion protein, the subject of this talk and my research, is actually roughly 200 amino acids. Now, each amino acid has its own property, chemical physical properties, and the combination of these properties then result in the final form, meaning the folding process and the final form of the protein. There is a paradox in the scientific community called the Levinthal's paradox, that basically if you take whatever kind of biologically relevant protein and you try to reconstruct all the movements that the amino acids should make to then lead to the final form of the protein, then you will come out with an astronomical number of possible conformations, meaning possible movements that then result in the final protein. So this is just to summarize, it's a very complex kind of field of science to understand how proteins really reach their final conformation. One help actually definitely comes from recent emerging computational technologies. And in fact, today it's possible, thanks to large supercomputers, to simulate the entire process of folding for very small proteins. Proteins that are, unfortunately, in the vast majority of cases, are not biologically relevant. To make an example, to simulate the entire folding process of the prion protein, it will take us 1 per 10 to the 14th microseconds, which are basically hundreds to thousands of years. So feasible, but practically unfeasible. Now, what I discovered once I joined the University of Trento is that sometimes it's much better to have a good colleague than a large supercomputer. In fact, this colleague of mine in the Department of Physics, Pietro Faccioli, actually invented a way of simulating folding of proteins in a very short time. So in the range of weeks to months. And then the result is basically this video that you can see here. That is the entire process of folding of the prion protein. This is the very first time that someone can reconstruct this entire process that you can see as a movie. Now, just for reason of time, I will get to the point. Along the process of folding of the prion protein, we identified an intermediate species, meaning a form of the protein that is transient, but still existing for a very short time, but existing, like a form of the protein that precedes the final state that we call the native state, so that the final form of the protein. Now, one idea then was that if we are able to target this transient intermediate state along the folding pathway of the protein with a drug, then we could actually stop the production of the protein, because we know that the quality control that every cell has will recognize that not completely finalized, let's say, form of the protein as an improper product, and then, you know, trash it out. So we tested this hypothesis, but then applying kind of more canonical computational and experimental techniques that are common in pharmacology. In particular, here we try to target a specific part of this intermediate form of the prion protein with, you know, lots and lots of drugs. We came out with 31 predicted molecules, small molecules binding in that pocket. And likely for us, one of these molecules, in reality 4, but I'm presenting data just for one today, one of these molecules was very effective in doing what we predicted. What you can see here, this black kind of stain signals, is nothing but the cellular prion protein that we detect with an antibody. And as you can see from left to right, the erasing amount of our compound called SM875 is corresponding to a difference in the signal of the prion protein, and eventually a drastic reduction, something that the graph below exemplify very well. So this protein is the first degrader, so a molecule that can block the folding and then leads the protein to degradation that we know so far for the prion protein. Now, few other data we know, and I'm just gonna, you know, summarize lots of data in few slides. We know that this compound is very specific for PRPC because we tried it on different proteins that are very different from PRP, but share many different, let's say, features, like the way that these proteins are expressed is very similar to PRPC. And to make a long story short, the compound does nothing to these non-relevant proteins. To summarize, again, a great deal of data, what we found was that our molecule is truly impacting the formation, the first, let's say, production of the protein during the folding process, so that the protein actually never reaches its usual location, which is the surface of the cell. And thanks to this property, then, our compound, incubated together with cells that are replicating prions, is able to really drastically lower the amount of prions that are replicating in these cells. So what we do now with this very promising compound that is acting in a completely novel mechanism, at this point, we try to go along with the classical, let's say, drug discovery pipeline, meaning that now that we collect direct information on the properties of these compounds in the lab, especially in cell cultured in our lab, now we're trying to make it better in order to be able to provide this compound to organism, mice first, and, you know, experimental models that we can handle in the lab, and then, if we're lucky, also translate that information to human being. So now we are exactly at this point, and thanks to the support of the CJD Foundation, we are making lots and lots of molecules that are very similar to the SM875, which we call the precursor molecule, but we test all the time. Whenever we make new molecules, we test their properties. And if we're lucky, we improve the properties, and at the same time, look for properties that are ideal for a compound to be provided in vivo, so in real organism. And I mean, I don't want to enter into the details, but we are reaching molecules. We already have identified molecules that are slightly better or even much better than the one that we initially identified, so the precursor molecule. And this is just recapitulating what we want to do. We want to fully explore the chemistry of this compound, and then also study what is called the pharmacokinetics, meaning how this compound may interact with a living organism, and then test its therapeutic potentials directly in animal models. At the end of this, you know, feedback loop, if we're lucky, we come out with a molecule that is administrable in vivo in human beings, and at that point we will enter, you know, clinical phases. We're still far away from that, but we believe this is a very good, you know, promising starting point. And I will just finish by thanking a large cohort of collaborators here from, you know, many different places around the world. And of course, our funding entities, the Telethon Foundation, that's basically sponsorized my lab, and of course, the CJD Foundation that brought me here today, and all the audience for the attention.